The rest of chapter 5 goes over I almost stepped on your foot there, Haley. Covers, I did, but I didn't crush it. Okay? So the rest of chapter 5 covers transport. All right? We talked about the structure of the cell membrane, and the structure of the cell membrane confers function, that function of selective permeability. All right? What goes in and what goes out. All right? What cannot enter the cell, what can enter the cell. All right? And the same thing, what can leave and what cannot leave. All right? There are certain processes for transporting material into and out of the cell. Okay? Some of them, which you probably have covered or listened to the videos, are passive, meaning that they do not require the breakdown of ATP. They do not require energy. All right? And some of them are active, meaning that they do require ATP. Okay? We're going to talk about a couple different types of passive transport. One example is diffusion. All right? We're going to talk about simple diffusion, which we'll see up in a second. We're going to talk in another example of diffusion called facilitated diffusion. All right, and we're going to talk about the movement, the diffusion of water, osmosis. Okay, but let's start with simple diffusion. So diffusion is the tendency for molecules to spread out evenly into an available space. All right, so we're going to start in some area, whether it be in air or in liquid or in whatever it might be, there's going to be a high concentration of something. All right? And eventually, what's going to end up happening is that those molecules will evenly disperse in the area you talk about. All right? So let's say it looks something like this. Here's a little beaker, all right, filled with water. And I drop a little tab of Kool-Aid, food coloring, inside that beaker of water. All right? Eventually, what's going to happen? Your beaker of water, well, let's finish drawing the beaker there, right, of water is going to have dye. That's pretty good. Evenly spaced. Right? It's going to start in some, you drop the dye in, and it's going to diffuse throughout the available space. Okay? You guys do this every day when you have your iced coffee. They dump the 6, 7 sugars in, right? And that sugar is concentrated, lots of sucrose molecules in one area. Right? She stirs it around, and they diffuse, they dissolve first of all, and then they diffuse throughout the entire water solution. Okay? Was any ATP involved? Did I say anything about it? No. No ATP is used. In this case, are there any cells involved? No. Okay? So diffusion is not a cellular only process. It happens in all aspects of, it's a physical process. It's a chemical process. Okay? Another example. How many of you enjoy brownies? Home-cooked brownies? Oh, yeah. Right? So, mix up a nice little brownie batter, stick it in the oven. Maybe 10, 15 minutes later, what does your kitchen smell like? It smells like brownies. Right? Why? But the brownies are in the oven. Why is your whole kitchen smelling like brownies? Diffusion. Okay? What you're doing is you're cooking, and as you're cooking the brownie batter, little chemicals are being released in the oven, and they diffuse from an area of high concentration in the oven to outside of the oven in the kitchen, areas of low concentration. All right? And that is the hallmark of diffusion. We're going to always move down the concentration gradient. Right? So right here, this is high concentration, and then out there, low concentration. Right? So whatever's going to diffuse, or whatever we're talking about, is going to move from an area of high concentration to low concentration. Okay? So here's our molecules of dye, right? Net diffusion. Eventually, what do we end up with? Equilibrium. Things are evenly spaced, all right? They're still moving, but they're moving at the same rate. There's not an area of high concentration versus low concentration. They are equal, okay? And you see this. This is showing you net diffusion. There's high concentration on this side, right? And so we move past the membrane because they can fit through by size, and you end up with equilibrium. Same thing happens here, right? You have one substance on one side of the membrane, another substance on the other. They will end up being or achieving equal concentrations on either side. Questions on that? So diffusion, no ATP, passive process. It depends upon a concentration gradient, an area of high to an area of low. That's the movement. All right, so we go through your concentration gradient. And this is passive transport because it does not require ATP. Questions on diffusion? Do you understand what passive transport is? We understand what a concentration gradient is. We have a concentration gradient in this room right now. High concentration, low concentration, right? Lots of students on this side, no students over there. Are we at equilibrium? No, because you're not evenly spaced out. Good job. Now, osmosis. Osmosis is defined as the diffusion of water across a semi-permeable membrane. Brett.
I just had a question about the equilibrium.
Mm-hmm.
If there is no movement, then is it still considered equilibrium?
There is movement. There's just no net movement.
Well, I'm saying like if it's stopped though. Like if the particles are stopped without the actual like movement, would it still be equilibrium?
If they are evenly spaced, yes. All right. Osmosis is the diffusion of water across your semi-selectively permeable membrane. All right. Now, water is a little funky, and osmosis can be somewhat confusing. Okay. Understand that the movement of water still follows a concentration gradient. All right? So, if I have our beakers again. Oh, not beakers. Let's just draw a big beaker. Nope. Get rid of that. We'll draw this. All right, here's our beaker. Okay? And here's our membrane. Say there's a membrane that goes to the two, and we have water on both sides. Okay? So now on this side, on side A, B, all right, on side A, we'll say that this is 70% water and 30% solute. Okay? And on the other side of the membrane, we're going to have 90% water and 10% solute. Which way is water going to move? It's going to move towards which side, A or B? A, right? Why? All right, because it has higher concentration of water, all right? It's also moving at the higher, so higher concentration of water towards A, because B is higher concentration of water than A, right? But water always moves towards the higher concentration of solute. Okay, so there's two ways you can look at this. You can look at it and to figure out the movement of water based upon how much water's there, okay, and it always moves from high water to low water. Or you can look at it opposite and look at the concentration of solute, and water always moves towards high solute. I think that's kind of confusing, because we've been just saying about diffusion of water, right? And we know diffusion depends upon a concentration gradient, right? And what's that? What's the movement? Go from where? Low to high, high to low, high to low, right? Now it's diffusion of water, and I say it's actually going to go low solute to high solute. That can be confusing. So I like to explain it as high water concentration to low water concentration. You're still using the same concentration gradient, okay? If you learned it previously opposite, it's still the same thing, it's just a different way of looking. All right, so here's your sugar molecules in water. What do you know about the sugar molecules in up here? Minimize that a little bit. In this part A right there. What can we say about the green sugar molecules? Right here. They're not evenly spaced. One side is higher than the other, yes? Right, so the right side is higher than the left side. So what's going to happen? Where, in terms of sugar, what wants to happen? It wants to go towards where, Carolyn? It wants to go towards the left, right? Sugar does. Why isn't the sugar moving to the left? The membrane is selectively permeable, and it's not allowing the sugar to pass by. Okay, the membrane is permeable to what though? To water. So instead, if sugar can't move, the motivation is still there. We want to reach equilibrium of water on both sides and sugar on both sides. So what ends up happening? What's happened? Water has moved from left to right. And now they're pretty evenly spaced, right? So now we have an equal amount of solute on both sides. All right, and if we look here, now when we reach, what is this now? What's happening here? These red and white Mickey ears are water molecules. Is there a concentration difference? Which side has more? The left side. So again, high concentration to low concentration. That's not to say water doesn't move back, but what's the net movement? It's moving right towards the higher solute concentration. All right, which leads us to what we call tonicity. Tonicity is a way of describing the concentrations of solutions. All right, so it's the ability of a surrounding solution to cause a cell to gain or lose water. All right, now cells are mostly made of water, yes? Thankfully, yes. Okay? But they're not 100% water, because there's proteins and carbohydrates and et cetera that's mixed in. All right? But an isotonic solution is a solution that is equal in the amount of water inside the cell as it is outside the cell. So there's no net movement of water. All right? Because water is moving across the membrane at equal rates, going in as fast as it's coming out. Now, tonicity is really looking at the solute concentration of solutions, okay? So now the other two here are hypertonic and hypotonic. These describe the solute concentration within the solution. Hypertonic, hyper means over, okay? Hyper means above or over, right, normal. So hypertonic means that the solution has a higher amount of solute than what is in the cell. So if it has more solute than what's in the cell, does it have more or less water? Has less water, so where's it going to go? So water's going to move out. And the opposite with hypotonic. Hypo means under, right? Hypoglycemia is low blood sugar, so hypotonic, the solution has a lower amount of solute than what's in the cell, a higher amount of water, so water rushes into the cell. Hypertonic, hypotonic. All right? Hippo, right? Hippo's big, right? So what's the cell going to do? Hypotonic solution, hippo is going to blow up, right, and swell. Here's your little diagram showing, right? When you put red blood cells in a hypotonic solution, water rushes in and it can burst. Isotonic, equal movements of water in and out. And hypertonic, there's a loss of water and you end up with a shriveled cell. That is very different than plant cells. Why? They have a cell wall to protect them, prevent them from lysing, right? So normally, they're actually in a hypotonic environment, so they are what we call turgid. They're actually pushing out against the cell wall. That's when you get nice crisp lettuce and plants are, standing up, whatever it might be, okay? If they're isotonic or even hypertonic, they lose water and they lose their, I don't know what the best word to describe, their turgidity, I guess, right? They lose their stiffness, okay? So, and then that's called placid, and if that even continues, it is plasmolyzed, meaning it's actually pulling away. If you see here, this is the cell wall, right? In this case, the plant cell is actually pulling away from the cell wall, which is causing a problem, right? Because it's losing water, all right? This is the situation that plants want to be in all the time. Okay? So, obviously, hypotonic and hypertonic solutions conditions cause problems for organisms, especially marine organisms or aquatic bacteria, plants, etc. All right? So they have evolved ways to regulate water amounts. This is called osmoregulation. It's controlling solute concentrations and water balance. Really, it's water balance, all right? Because a lot of times you're not going to be able to move stuff that's inside the cell, it's in solution, out of it, but you can always regulate how much water is inside and outside. And the paramecium and other aquatic single-celled organisms do this by bringing water in or spitting it out and having what we call a contractile vacuole that can pump water in and out that will allow it to regulate tonicity across the membrane. There's an example of a contractile vacuole, right? So it's an opening inside the paramecium that will help regulate the amount of water outside and inside. It will swell and contract in order to maintain water balance. All right, we went through flaccid, and so you see here
Okay, so the flaccid one's just like more,
So this is pushing out, and this is kind of like, it's just normal.
okay,
All right, so, middle morning, you water your plants, okay? In the garden, right? Hot day comes, by the afternoon, what ends up happening to them?
they start to wilt.
They start to wilt a little bit. This is the wilting part. They're not like this yet. You let them go another day, you're in drought, they start to look like this. All right? Then you add water, it sucks up, right? And it's transported, and you get back to this state. And then the plants become stiff and non-wilted. Okay?
Thank you, sir.
So now facilitated diffusion. Ooh, it moved on its own. Okay? Facilitated diffusion. It's still diffusion, so it requires what? A gradient. From what? From high to low. Okay. Unfortunately, there are some things that the cell needs that do not pass the membrane. Glucose is one of them. All cells need glucose, okay? But glucose is too big to pass through the membrane on its own, all right? So the proteins that are inside the plasma membrane form a channel or a carrier to help assist the transport of those big molecules or charged molecules or whatever else doesn't pass the membrane on its own, okay? But it's still diffusion, so is there ATP going to be utilized? No. So this is just utilizing a protein carrier to get something across the cell. Okay? Usually it's something that is too big, charged, it's a polar molecule, it can't pass through the lipid bilayer on its own without causing problems, so we evolve these channels to allow it to occur like that, right? Water is one of them. Water is a polar molecule, correct?
Yes.
Yes. Do polar molecules get past the plasma membrane easily? No. Now, water's small enough and some of it does pass the lipid bilayer a little bit, but a lot of it goes through a channel called an aquaporin. Okay? So, really, osmosis is a type of facilitated diffusion. So this is what it looks like. Okay? Here's two examples. A channel protein, like an aquaporin. So there's basically this protein which is embedded inside the plasma membrane, and it forms a tunnel, which stuff can go through. All right? And that tunnel is regulated. It's like there's a toll booth. It's not just, hey, just everybody come through and let things go. But high concentration outside the cell, lower inside the cell, but it can't pass through the bilayer. It has to go through the channel. Same thing with the carriers. High outside, low inside, and that ligand or that substance will bind to the protein, cause a change in shape, and then that protein is released into or out of the cell. All right? This is extremely important for charged molecules, right? Ions that move in and out. Yes?
How do those not use energy if they're like moving and stuff?
The driving force is what? The concentration gradient, all right? So just because this is binding, right, the binding actually causes the shape. And if there's not a lot of it out here, it's eventually going to stop, right? So this moves it in, and this protein will bind another one and move it back out. Equilibrium. The driving force of this is still the concentration gradient, right? We're not utilizing any ATP here, all right? There's no work that has to be done. It's already set up for us, okay? So it's a subtle change, and it's usually by binding and release. And again, no energy input is required. Questions on passive transport? Diffusion, osmosis, facilitated diffusion. three types of passive transport. Okay? The last part of the chapter is active transport, which means what? ATP is utilized. Okay? And typically, we use ATP to move substances against the concentration gradient. So we're going to go from an area of low concentration to an area of high concentration. Okay? And most times, this also requires a protein carrier that's involved. So it's like facilitated diffusion, except the proteins are involved. We're going to utilize ATP in this case that will move substances in different ways. All right? So active transport requires energy. Now this says usually in the form of ATP. Usually. Right? The reason why we say usually is that sometimes there are actually two types of active transport, primary and secondary. All right? Primary active transport is probably what you've talked about and you've learned. It is utilizing ATP to move a substance across the membrane. All right? Done. However, secondary active transport is sometimes we push a substance across the membrane, say an ion, a hydrogen ion, utilizing ATP, right? And now, when we do that, we actually now we have a lot of hydrogen ions outside the membrane as opposed to inside the membrane. What is that? What have we made? So, here's my cell. Here's my carrier protein, and I'm going to push a bunch of hydrogen ions out here. So, lots of these guys out here. Now what? And I've used ATP in the process, so ATP is broken down to ADP. Now, what do we have out here? The concentration gradient, right? I have lots of hydrogen ions outside, and what do they want to do? They want to get back in. Okay? So now if I had another protein out here that was regulating the movement of these hydrogen ions back in, I could also co-transport something in with it. So the movement of hydrogen ions, not using ATP, right, as it goes past, as it moves something back in, that is secondary active transport. Why is it secondary? It's not directly breaking down ATP, but it depends upon the primary active transport of moving the hydrogen ions out. Okay? So usually we break down ATP, that's primary active transport. Yeah, we could stop there. But there's another sub-level to that where this concentration gradient of hydrogen ions or substances, whatever, that's produced by the active transport can actually move things in as well. So we have primary, and this is secondary active transport. Yes?
So is it because we did use ATP at one point, but just not?
Directly, just not directly. All right, so this is secondary active transport because ATP was used at some point to produce this concentration gradient. All right, and then we use that.
I'm confused as to why they would go out and then back into the cell.
It's a good question. Seems inefficient, right? It seems inefficient. But if you depend upon this, okay, to drive another protein, then that's why it's done. Okay? It seems inefficient, but it actually is used. A lot of times these proteins can't do two things. They can bind something that's going to be pushed in, but it can't hydrolyze ATP. See what I'm saying? So this allows something to happen that the protein doesn't have the function for. Other questions? Yes?
What type of stuff is the hydrogen bringing back into the cell?
Sometimes it's like large molecules, glucose. It could drive ATP synthase. Whatever the function is what's called a proton motive force. All right, it's a concentration gradient that's going to cause this protein or allows that protein to do other functions. One of the best examples of this is the sodium-potassium pump. All right, and this is present in all your cells, which keeps your cells at what we call a resting membrane potential. All right, there's an amount of positive ions outside the cell versus inside the cell. So this is what it looks like. One protein is going to transport sodium and potassium, ions. Now, why don't ions move across the plasma membrane on their own? Because they're charged. All right, awesome. Okay? And ideally, the goal of the pump is to have more positive ions outside the cell than inside the cell. So to do that, we start at number 1. Okay? Sodium is high inside, low outside to start. So we're actually going to bind 3 sodiums, bind to the protein. Okay? 1, 2, 3. And at that point, ATP is hydrolyzed. The binding of the sodiums did not change the structure of the carrier, did it? Did it change the structure of the carrier? No. All right? So this is not facilitated diffusion. If this was facilitated diffusion, the binding of the sodiums would change the structure of the protein, and then the sodiums would be kicked out. But we require ATP to change the structure of the protein. Okay? When that happens, now that protein does no longer like sodium as much, releases them to the outside of the cell. In this structure, once sodium is lost or released, potassium now binds to the cell, or to the protein. Potassium binds, and that causes the protein to return to shape, and it loses that phosphate. It returns back to its original shape, releasing potassium into the cell, and then back to the same point. Again, what you see here, in this case, sodium is low inside, and potassium is high inside. Right, you see right here, and then outside. So the movement of potassium is going from where to where?
Outside to inside.
Going out to in, and what is that? Going from low to high, against its concentration gradient, right? And the movement of potassium is going from low to high, against its concentration gradient. This is the stereotypical example of active transport. We're moving substances against the concentration gradient, okay, while hydrolyzing ATP, okay, active transport. And it requires a protein carrier and hydrolysis of ATP. Questions on this? All right? Eventually, what's going to happen? I'm moving 3 sodiums out, 2 potassiums in. And this continues to go over. This cycle just keeps going over and over and over. What's going to end up happening outside the cell versus inside the cell? I'm going to have a lot more sodiums outside the cell, right? But what? What else? It's going to be more positive. I'm putting 3 positives out, 2 positives in. 3 positives out, 2 positives in. Every cycle, I'm adding an additional positive ion outside of the cell. Okay? So, in this case, the cell will always be more positive outside than inside. And that's what we call a resting membrane potential. Okay? So we have lots of positive ions outside, less positive ions inside, so it's a negative. So if I open all the channels for sodium and potassium, what's going to end up happening? What's here? Sodium's high. What's going to end up happening? I open all the channels. I just like open all the gates. Now sodium can move in on its own.
It'll flood the cell.
It'll flood the cell. It's going to go down its concentration gradient. Okay? Sodium will rush in. Potassium then starts to rush out. And now we have to reset the whole system. That happens every day in your cells. Every day. Right? Every time you think, I'm going to write something down. Every time you feel a nervous twitch. All the times right now that you're taking in all this information of sight, sound, right? It happens in every neuron, every muscle, all the time. When sodium allows to be rushed, so nerve action potentials, muscle twitches, that is all based upon this right here. It's all based upon setting the amount of positive ions outside the cell higher than what's inside the cell. So now that cell can be stimulated to do what it needs to do. All right? Go through these. All right, so let's compare passive transport versus active transport, and this is the big part right there. Okay? Now, the sodium-potassium pump is also an example of co-transport. If you're cohabitating with someone, what are you doing? Living with someone, right? There's more than one person living together, okay? All right? So if it's co-transport, what is happening? More than one substance are being transported, okay? And they can be either the same direction or opposite directions. In the case of sodium potassium, we have opposite directions, right? That's what we call antiport. Two substances are moving in opposite directions. Okay? If two substances were moved together, that would be what we call symport. Co-transport of two substances in the same direction. So glucose and a hydrogen ion are moved into the cell. That is symport, right? Because they're both going the same way. Questions on any of this? Diffusion, facilitated diffusion, active transport up to there. And look at that. We already talked about membrane potential, the voltage across the membrane. Yes, Kid.
What did you call it? It was the resting.
This is the membrane potential. It's up there right now. Membrane potential, the voltage across the membrane. Right, and that's created by, so it's resting membrane potential. What you end up with and what you've produced is what we call an electrochemical gradient. All right, so we have it's basically a concentration gradient of ions. So here's your membrane, right? We have lots of positives out here, only a few negatives, right? And we have maybe less positives in here. Okay? So what do the positive ions want to do? They want to flood in. That's what it boils down to, is there's an electrochemical gradient. There's a concentration gradient of ions, more positive on the outside versus the inside. All right? And what that does, it produces a chemical force. Because what do the ions, the high concentration of ions outside want to do? They want to rush in, right? They want to move down its concentration gradient. And cells can utilize that to produce ATP, to move substances, right? So this electrochemical gradient can be produced by active transport, and then cells can use this in secondary active transport to move things in and out. All right. So, an electrogenic pump. All right. We've talked We basically already talked about one of these. The sodium-potassium pump, right? Pump means it's going to move things across the membrane, right? In this case, an electrogenic pump is going to move hydrogen ions, all right? The sodium-potassium pump is an example of an electrogenic pump. It's going to produce a concentration difference or an electrochemical gradient across the membrane. And there's also what we call proton pumps. They're going to move hydrogen ions, all right? And while the cell is producing that concentration difference, it's producing what it can utilize as for work, for cellular work. So that concentration gradient can be utilized to do some type of work, whatever it might be. All right, whether it's to produce ATP, whether it's to move things in and out of the cell, it's some type of cellular work, usually some type of transport. All right, so there's your proton pump. Going to utilize ATP, push hydrogen ions across the membrane, all right, and then you're going to use that. In this case, this we call a proton motive force. All right, so this, what they end up want to do is they want to go back there. That's a proton motive force. And in this case, let's say there's a protein out here that will take glucose and move it through, but only with a hydrogen ion. Okay? So in this case, if glucose wants to get through, it needs this concentration gradient of hydrogen ions outside the cell to push it through. Right? This is secondary active transport. This is co-transport, right, with the hydrogen ion that moves through, and it allows for the cell to do these things without utilizing too much ATP. Okay? We went through co-transport. Don't add anything. Right, there it is. Look at that. Know what I'm talking about, huh? Done this a couple times. So there's your proton motive force. It's going to produce this hydrogen ion concentration. Hydrogen ions are going to bind to this protein. You got a sucrose co-transporter going to bind and move the sucrose into the cell. Okay? So this is your primary active transport, right? And then the sucrose right here, that is your secondary active transport. It's secondary because it depends upon ATP being utilized in the first place. Yes?
Does it matter that sucrose and hydrogen, the co-transporter, they're like paired together? Is that like significant?
That is significant. Okay? So the question was, before you start wrapping up, is it significant that they are paired together? Yes. Okay? If this is not there, if the hydrogen ions are not there, the sucrose can't bind to the transporter. Okay? or it doesn't change shape and it doesn't allow it to get inside the cell. Karen.
Is it only hydrogen ions that can use?
It's not only hydrogen ions. This is just showing an example. We can use hydrogen ions. We can use sodium, potassium ions, other ones, calcium ions. But this is going to be important as we move forward with photosynthesis and cellular respiration as well. Sam.
So ATP is broken down to ADP during primary.
Yes.
Can sucrose bind if it's like not hydrogen but another one? Or can sucrose
No,
only work with hydrogen?
So this will only, it depends upon the protein, but it'll be specific for the ion. So in this case, so this is sucrose hydrogen co-transporter. It'll only transport sucrose and a hydrogen ion, not a calcium or anything like that. Okay? Other questions? All right, your quiz on Friday, you have your study guide is posted. Cardone? Yep. I like Carbone better. Okay? We're going to finish chapter 5 today, and we're going to start chapter 6. Okay? Because I'm behind the other classes, so we got to catch up. Got it? Good. So chapter 5 finishes up transport. Okay? We went through passive transport. What are examples of passive transport? Diffusion. Osmosis. Facilitated diffusion. Okay, good. Active transport. Oh, yeah. Oh, what about ATP? It's not just word association. It uses ATP. There's a sodium potassium pump is an example of active transport. Okay? And we talked about primary versus secondary active transport. All right, but just know for active transport, usually it's you can pump things against its concentration gradient and also utilizes or hydrolyzes ATP. One other example of active transport is bulk transport. Okay, bulk transport. This is like going to BJ's and we're buying the big thing of paper towels. Okay? We're having 32 rolls of paper towels and we're going to keep them for a year. Okay? We're going to get rid of all of this stuff in large quantities. Right? It's not just a couple ions that are passing through. This is a big bulk delivery of proteins that can't pass through. Okay? So we're going to move things in and out of the cell in large quantities. Okay? So these large molecules such as polysaccharides, big proteins, right? They move across the membrane. They really don't move across the membrane. They are shuttled to the outside of the cell, okay, in vesicles. And so this is bulk transport. All right? So what's going to end up happening here is we're going to have our cell. Okay? Here's our cell. Our cell is going to make some type of protein. All right? Some type of protein. But that protein is going to be made inside a portion of the membrane, a vesicle. Right? So that green circle up there is actually really a phospholipid bilayer. It is this made up of the same stuff that the plasma membrane is made up of. Okay? Okay with that? There's proteins in there, there's phospholipids in there, all right? So it is the same stuff, the same contents, components of that you'll see in the plasma membrane. All right? So the proteins are packed inside of there, and eventually what's going to end up happening is that this vesicle will move to the membrane, right? And it'll actually fuse with the membrane. Right? Apparently we can't do that today. All right, so it's going to fuse, and we're going to have the proteins eventually are going to be released. Okay, so it'll end up looking like something like this. The proteins that were inside there are now going to be released inside the cell. As you end up with the vesicle fusing with the plasma membrane. Okay? You've seen oil in water before, yes? Okay. So now you mix up the salad dressing, you mix up the water, and there's all these little oil droplets, right? Lipids inside water, basically what a plasma membrane looks like. And if you let them sit for long enough, they start to collide, right? When they collide, what happens? One oil droplet joins to the other oil droplet, because due to the hydrophobic nature of it, right? It spits the water out. And then they collide, and that's the similar thing that's happening here. The phospholipid bilayer of the vesicle is going to fuse with the phospholipid bilayer of the plasma membrane. And therefore, the contents of that vesicle will be released out of the cell. This is exocytosis. All right, exo means out of the cell. So what we've drawn here with our cell and our vesicle, our proteins in our vesicle, right, that then move to our cell again, okay, our vesicle now to the edge of the of the membrane, and eventually fusing with the membrane and emptying the proteins to the outside. That is exocytosis. Exo means out, cyto means cell, so out of the cell. All right? Where is ATP being utilized here? Brett.
Push the vesicles to
The-
the side of the membrane.
The movement of the vesicles. Remember when we talked about the cytoskeleton and the molecular motors? The cytoskeleton is a pathway. It's a railway system for movement of these vesicles in and out. So we have little cars on the tracks that are pulling the vesicles, pulling their cargo. And there's some type of energy that has to be used to move them in and out. Right? That's where ATP is hydrolyzed. Okay? So the movement of this vesicle from inside the cell to outside the cell requires ATP to be hydrolyzed. So if things can get out, big things can also get in, right? So there's this two-way street, okay? So we have exocytosis and we have endocytosis. Endocytosis in humans is important for white blood cells engulfing bacteria. All right, and there are 3 types of endocytosis. We have phagocytosis, which is cellular eating, penocytosis, which is cellular drinking, and receptor-mediated endocytosis. Okay? So phagocytosis, this happens in amoebas. All right, I'll minimize that. You guys can still, let's make it a little bit bigger so you can copy. All right, here's our amoeba here. All right, nucleus. And then there's this little bacteria out here that this amoeba wants to eat. Okay? So what's going to end up happening is that this amoeba is actually going to engulf the cell. All right, so it's going to grow out and around, eventually forming this food vacuole inside. Okay? So then it'll kind of grow like this, eventually going to that point where it's inside. Okay? That is phagocytosis. Some type of solid material, debris, a bacteria, whatever it might be, is engulfed by the cell. Right? The cell basically grows around it or moves around it and takes it in. Note, it is now a vesicle. Right? There's a chunk of the plasma membrane that's engulfed, that's encircling this stuff. Okay? If that stuff that was taken in is just liquid, that is penocytosis. All right? So some type of solid material is phagocytosis. Any type of liquid is penocytosis. And then finally, we have receptor-mediated endocytosis. Okay? Receptor, what's a receptor?
Something that like
Receives some type of signal, right? All right, so if it's receptor-mediated, what does that mean to you? Cardone.
That the receptors send a signal saying we receive something, and then they do a vesicle.
So in order for endocytosis to occur, that receptor has to have the signal first, right? And that's exactly what happens, okay? So in receptor-mediated endocytosis, the receptor is bound to its signal, and then that receptor or part of that membrane is brought in as a vesicle, okay? Receptor-mediated endocytosis are ways which viruses and bacteria can infect cells. It's also ways where the cells of your body can regulate response to hormones. Okay, say there's this, say there's a cell in your body, okay, and it has a receptor out here that's going to bind to this hormone. Okay? And the hormone binds, and the hormone keeps coming. The signal's there. Okay? If the cell is no longer gonna be desensitized to it, what's gonna end up happening? That receptor and the ligand gets internalized. Okay? Now there's no more receptor on the membrane, no more response to the hormone. Okay? That's an example of receptor-mediated endocytosis. Okay? So here's what it looks like. All right, phagocytosis, pseudopodium. Okay? That engulf something. What?
How did the other two get triggered to start then? Or is it just any time that something like
So there is a trigger, okay? Think about where the trigger comes from, okay? And usually in phagocytosis and pinocytosis, the trigger is cellular derived. It comes from, the signal's coming from inside the cell to go out and form the pseudopodia and engulf something. Same thing here, okay? And in this case, there's some pitting, that's some proteins accumulate and it's brought in, but it's a cellular derived, originated signal from the cell. Here, the signal is actually from outside the cell, okay? Receptor-mediated endocytosis is a response, it's a reaction to something on the outside. These are cellular actions, all right, instead of a reaction. Good question. Yes?
So a food vacuole is only used in phagocytosis?
Yeah, oh, well, yes. You could consider that once that's brought in, that vesicle is now a food vacuole.
And a vesicle too?
Yes,
Like, okay.
in that case, vesicle and food vacuole are synonymous,
Okay.
okay?
All right, yeah.
So here's an electron micrograph of an amoeba engulfing a bacterium, right? So this amoeba here is going to grow out, and will continue to grow out till it surrounds this, and then it will digest the bacteria, all right, by using hydrolytic enzymes that are dumped into the vacuole or the vesicle, and that break down whatever was brought in. Okay, and here's pinocytosis, right? In this case, the cells are forming your vesicles. All right, and then last, your receptor mediated. Okay, receptor mediated is a little different. Okay, you'll see here there's some type of coat proteins, right? You have to identify which part of the membrane is going to be brought in. Okay, so the proteins identify that part, and then it starts to come in, and eventually forming what's called a clathrin-coated pit. Right, and there's a lot of intricacies about this, about where things go. Okay, so it's not just willy-nilly in that when we produce, the cell's going to produce proteins by exocytosis. It's not just, hey, just go to any part of the membrane. I don't care where you go. There is a direction, okay? And there are protein interactions that identify, there's like a protein interaction between the vesicle and the membrane that stimulate that opening between, or the fusion between the vesicle and the plasma membrane, right? But that is, that's a cellular physiology lecture, not necessarily a bio lecture. Okay, questions on that? Okay, good. So I'm going to render this. Any questions on anything with, what's today? 10, 21, 16.